Dr. Michael Schoof, how are you, sir?
I'm doing good. How are you doing?
Doing all right. Thanks for being here. I've been excited to chat academics with you for a while, and I'm finally
Yeah,
glad
well,
we get to do this.
I'll do my best to live up to the academic goals then.
Oh, no. I mean, I think that it'll be fun to hear your perspective about how you navigated academia, especially the field that you're in, which I think is of profound importance and is going through such important changes. And I think the effects that we're going to see as, AI starts compounding some of these discoveries and accelerating some of these, some of the solutions and innovations and getting them to market faster. I think the world is about to see new kinds of relationships with their biology and the sort of potential new treatments and potential new things in the world. I wanted to start with your 2020 paper on the nanobodies and COVID, because that stood out to me as a remarkable and topical and timely paper, leveraging your research that you could tell was years in the background in the making, but had sort of like a lightning moment to be used for the most critical problem of the time. And I just sort of wanted to unpack with you what that was like, what it felt like to hold the lightning for a little bit and go from there.
Yeah. Well, that was a very wild time for everybody, obviously. I think it's worth noting that I am not a virologist. I didn't train as a virologist. I trained as a biochemist. And so the way we ended up getting into this was mostly due to the fact that when lockdown started, all research at UCSF that was not COVID related shut down. And so there was a huge amount of manpower with a lot of expertise that was sitting idle or was going to sit idle. And so I talked with my professor, Peter Walter, and our collaborator, Ashish Menglik, and we basically said, well, we're sitting at home. Why don't we try to think if we can, do something that would be of use? And for entirely unrelated reasons, Ashish had developed this technology in grad school and then optimizing it when he started his own lab for generating nanobodies, which are basically small antibody fragments, which are derived from camelids. And they had this system that they developed for rapidly testing and generating nanobodies that could bind to whatever you wanted them to bind to. And they had been using it, and I had been using it as well, for totally not virology related reasons. So we were interested in, Ashish's lab is predominantly interested in using these in order to stabilize specific conformations of proteins so that they can then do structural biology on them. And I was interested in using it so that I could specifically stabilize different oligomeric states of the protein that my PhD is actually in, which is this protein called EIF2B. And that's where the vast majority of my papers and time are on. And so for totally unrelated reasons, we were using this technology. And then we all came together and said, oh, okay, well, our labs are good at purifying proteins, and our labs are good at making nanobodies, and they're good at solving structures, and they're good at doing cell biology assays and all these things that would be, in theory, useful and easily applied to COVID work. And so we put in a little short grant that got us a little bit of funding with a really quick turnaround time. And at first it was me, my roommate, and Ashish who were doing work. And then when it became obvious that we were moving in a positive direction, we were really able to expand and get more people from our labs involved. And it became a massive team effort. I mean, the paper is a 5 first author paper. I think there's 60 people on the paper in total. We ended up collaborating with groups in other countries. We were working with groups at Berkeley as well, multiple groups at Stanford. So it was a really great experience to get so many people together working on a common goal. And as the project evolved, I was, spending a lot of time in the lab, but also more and more time mostly coordinating the big picture stuff. And so as it was my first author paper, so it was quite the wild thing to have as my first paper. So I feel like I learned a lot from it.
Well, and I saw as much from your Google Scholar profile page that it was your first author paper, but it struck me. So this is November 2020, like right in the middle of it. It's like, we're still figuring out what reality looks like.
Yeah.
And, just the title itself, using nanobodies to essentially halt viral replication because you're binding to the spike protein. Is that right?
Yeah. Well, so the logic was, we want to make something that can bind to the spike protein and basically stop the virus from entering cells. Mm-hmm. And there's a decent amount of, precedent for this from what your body does naturally and what companies will do when they generate monoclonal antibodies for therapeutics. We took a different approach that a couple other labs have taken of trying to do nanobodies as opposed to monoclonal antibodies or other binders. The rationale for this is they're small and they can be produced cheaply and at scale, not necessarily in human cells, but they can be produced in yeast at scale. They can be dried down, aerosolized, inhaled. Delivery is, in theory, easier than monoclonals, which need to be infused. The issue that we encountered when we got to we want to commercialize this and get this actually into people was that there's very little existing pipelines for manufacture and distribution of these sorts of things because there's not a lot of precedent. There's a lot of precedent for monoclonal antibodies. There's not a lot of precedent for nanobody treatments. When I was doing this work more thoroughly, I think there was one nanobody treatment that was on the market. Don't know if there are more since I was heavily involved in this work, but even if there are more now, it's not common. And big biotech companies like existing workflows that are tried and true, even if the potential base of people that could use it isn't high. I can only imagine,
after the paper is done, then sort of the all the layers of approvals from FDA, from like getting supply chains up and running, to getting distribution deals, like the type of things that the pharmaceutical companies do at scale. There's a whole different thing going from scientific innovation to actual mass distribution. And I can only imagine what that world looks like and all the complications there and all that stuff. But it also seems that you need these kind of innovative sort of like lateral thinking around these type of bigger problems so that you can actually start building a platform that then, you build upon, right? Like, I think this was mRNA 20 years ago or 30 years ago. It's like, okay, there's no there's no mRNA vaccine, but there's like a platform here that we could use to explore all these kind of, it's a new delivery mechanism for treatment. And I think something like this is really amazing too. Why don't we talk about a little how you got there to the point where essentially your biochemical training, your academic path, and the research area that you're in now.
Yes. Well, one other quick point on the COVID work that I'd like to make, which is that, and this is more a plug for academia in general, which is that the initial outbreak of SARS was in the early 2000s, and then it, disappeared. It was no longer in the human population. But for 15 years, there were a lot of labs studying the first SARS and studying related coronaviruses. Labs like Jason McClellan's lab at UTA. And the fact that these labs were doing this research for the last 15 years, and that it was funded and supported, was what gave us such a head start to actually be able to know how do you design vaccines and how do you solve structures of, spike proteins from these viruses. So I just want to give a shout out to, those groups that, were out there doing work that a lot of people weren't necessarily that interested in for many, many years, but then became profoundly relevant. And I think that's one of the most important things about academia, which is that people being able to follow things that they think are interesting and that they think are important, even if they're not necessarily immediately commercializable, because down the road, it can become quite critical.
Mm-hmm. Amazing. Yeah, absolutely.
And I mean, I guess I'll use that topic of serendipity to segue into my answer to your question, which was you. It seemed open-ended. You were asking me, how did I get from somewhere to somewhere? Is that correct?
Roughly, yes.
Roughly. How early in the somewhere should I start, and how late in the somewhere should I end?
Well, how about like I'm an impressionable young undergrad deciding your major, deciding, the menu of possible futures as a freshman is gigantic, right? Like you could go into academia, you could go into industry, you could go into the arts, you could go into something new and weird. And I think that the sort of like when we very briefly synced up about a month ago or so, or 2 months ago, it was like, so I know what you're working on now, which I think is one of those things that is going to shape humanity. And I'm excited to get to it. So I'm excited to hear the path and the decisions that you made that got you to now a position where you have that academic freedom to explore this thing a bit more, let's say, leeway to, take creative risks and things like that. So I don't want to spoil where we're at today, but I want to hear how you got there.
Yeah, well, I might spoil it immediately, which is that from a very early age, I have been fascinated with science, and I think my parents would agree. From 6, I knew that I wanted to be a scientist. I don't think it was clear at that early age what kind of scientist I wanted to be. At that age, it was just a general fascination and love for the world. I would go to these mad science classes, and my mom likes to say that she would drop me off and I would squeal in excitement. And I made, at like age 5, I made toothpick models of atoms, and my mom's on video asking me like, what is this? And I go, it's an atom, silly. Don't you even know? So I was being a little smug back then. But I was very excited about science. I'd watch and about the future. And I would watch, Star Trek with my dad, and I would really just imagine how we can change for the better, and how there's the possibility for, technological advancement that can then drive human advancement and make people's lives better. And so that was kind of from an early age, the general thing that I was raised in, of just being excited and interested in science. And I think also from an early age, the thing that kicked me into being interested in biology and what I do now, which is aging research in particular, is watching people I know get older, and, to be honest, being scared of what that means. So being scared of, on a personal level, and for them, being scared of, age-related neurological disorders, being scared of dying. These types of really basic human feelings were a big driver in me thinking, okay, well, I'm excited about science. I'm excited about these things that I view under a microscope, and I'm excited and frankly a little bit scared about this big thing that happens to every biological system. And in high school, even, that was what I said to myself that I want to study. And so I came to Stanford with that goal in mind. And there were periods, where I would say that I did doubt whether that was what I wanted to do. Or where I thought, is an academic route the way that I want to go? Or where I thought, if biology actually was the thing that I should focus on, or if instead computer science is the thing that I should focus on. And if, completely leapfrogging a biological system and going into, computers to tackle this stuff. And, and we weren't really thinking about AI that much at the time, but these sorts of topics, instead of looking at it from a biological standpoint. So this general problem and interest in aging has always been there, but I would say that there have been times where I've thought, what is the right path to tackle it? Yeah. And so during the end of undergrad, and I worked in a lab at Stanford, an aging lab at Stanford, for all my years there. And at the end, I was, saying, okay, well, I'm going to go to grad school. And I ended up going to Cambridge for a year because I wanted to live in the UK. And then I ended up going to do my PhD at UCSF, in no small part because I really enjoyed the Bay Area and wanted to stay in this community. And then this is where I sort of had a bit of a decision making, which was, okay, I'm doing my PhD. This is going to be the first time that I'm, working for many, many years full time in a lab. I worked full time for 1 year in my master's, but it was a temporary thing. This is like, I'm going to be here permanently. What do I want to do? And I rotated in a couple different labs. One of the labs was this guy, Salvieta, who is fairly well known because of his work in the lab of Tony Wyss-Coray as a grad student, where they did these parabiosis experiments where they sewed a young and an old mouse together, and they saw that the blood from a young mouse could help rejuvenate the old mouse. And this is how you get Silicon Valley talking about blood boys and all that. And I rotated in this other lab, Jonathan Weissman, and another lab, Peter Walter, who does cell biology and biochemistry. And I actually made the decision at the time that I did not want to join the hardcore aging lab. I instead wanted to get a very basic fundamental understanding of cell biology and biochemistry. And that was driven by a genuine interest in those topics, but also because, and this is where, real world things come into play. I really did not enjoy the mouse work. It was quite unpleasant to do it, and I wanted to put it off as long as I could. And I was very excited about instead learning basic biology during my training. And so that's how I ended up in Peter's lab, where I decided, okay, I'm not going to necessarily study aging right away. Instead, what I'm going to do are study the basic core processes that go awry during aging. And then in later training, I can come back and have a more aging focused type of research, but only after I have gotten this really core fundamental understanding of, how do proteins get made? What are the conditions under which the types of proteins that are getting made change? And how does this mechanistically happen? And so that was sort of my mindset. And looking back on it now, I'm quite happy with the decisions that I made.
I think it's awesome. I mean, the way if I read it back to you, it's sort of like you took you went a rung lower into the ladder of abstraction to understand sort of the processes at a more sort of like at a reductionist level, where you're sort of at the chemical level trying to understand what's going on.
Yeah.
And I think that's awesome, because I think that it's easy to make I think it's hard to get the picture 100% right if you don't have the full stack understanding of everything that's going on. Which leads me to my next line of questioning. That is, okay, fine. Now we get to reveal that we're talking about aging, which is something that's been so present in my life for the past maybe 2 years. I think triggered by mostly COVID, now is the time to consider that, wow, sort of like we're at a stage in life where we're not kids anymore. And we have to navigate life as adults, see the generation above us getting a little older, see the generation above that getting significantly older, and starting to deal with some of that. And sort of okay, but what do I do about it? And so there's this pop culture flavor of anti-aging stuff, the Brian Johnsons and the Peter Attias and the David Sinclairs of the world, and the supplementing and nutrition aspect of it. And I think there's value to some of that. I think that there's I think that aspirationally being oriented toward preserving youth and toward preserving health is a good goal. But then I always wondered, but what's the actual scientific definition of this thing? Because as far as I understand sort of a biological system, it's all right, you're a transcription machine. You have a DNA sequence, and which to some order or another is just interpreting itself and replicating cells using that instruction set. And as you do this over time, the quality of the copies starts degrading, and the ability for the cells to retain their form, or their sort of ideal state corrodes over time. But I don't even know how you begin to quantify that. That's an intuitive definition that I have. So to the aging expert in the room, how do you and science in particular define aging?
Well, I think I think what you said is perfectly serviceable for us to have a discussion on. I think it's hard to put a tried and true definition on it, but I think in the general way that we think of it as a sort of a time-related deterioration in quality and ability to perform various tasks is, I think, more or less a fair working definition of what aging as we generally understand it is. I think all of us also intuitively understand aging on a macro level. Mm-hmm. And so my, interest as a scientist is trying to understand it on a micro level. And one of the things that you touched on is a good segue of, us basically being sacks of DNA that need to, replicate and then make machinery that helps us replicate and helps us perform tasks. And so anybody who has taken a high school biology class has heard of the central dogma, where you get from DNA to RNA to protein, and the proteins are the machines that actually do the vast majority of the work in running a cell. And so in grad school, I was focused on how do those proteins get made. And then in my postdoc, I've become very focused on trying to understand the diversity of proteins and how that changes with age. And so if you'll allow me a minute to give a brief overview of the complexity with which we're talking about, and then I can get to the topic of how do you actually quantify this. You have DNA, and 20 plus years ago, we sequenced the human genome, and we thought that there were going to be more genes than there actually ended up being. So there ended up being some 20,000 genes in the human genome. And those bits of DNA need to get turned into RNA. And then you have, once you've turned that DNA into RNA, you have a little bit more diversity. Now, all of a sudden, instead of, talking about 20,000 genes, you're talking about maybe 100,000 RNA transcripts. Mm-hmm. And then those RNAs need to get turned into proteins. And so you would think, oh, okay, well, if there's 100,000 blueprints, then there must be 100,000 unique proteins, but it actually gets much more complicated. And it gets more complicated because each protein can have different proteoforms, different post-translational modifications can be added to it. And so even though you're thinking, oh, okay, well, maybe there's only 100,000, there should only be 20,000 proteins, but between alternative splicing and post-translational modifications, you're actually talking about millions of different proteoforms that can exist in a cell. So you can have things like phosphorylation, you can have acetylation, you can have what I study, which is glycosylation. And so you can have all of these different small molecules appended to a protein, and they can dramatically change what that protein's function is, what other proteins it's binding to, what part of the cell it's in, whether it's enzymatically active or inactive. For example, in grad school, I studied this pair of proteins called EIF2 and EIF2B. And normally, EIF2 is the substrate of EIF2B. But when EIF2 becomes phosphorylated, all of a sudden it becomes the inhibitor of EIF2B. So a single post-translational modification can dramatically change how that protein is interacting with another protein. And so when we talk about, trying to understand aging, what I've sort of coalesced around being really interested in is trying to understand the diversity of proteoforms and how that changes with age. Because at the end of the day, the things in your body that are doing the work are the proteins. And if you have the best blueprints in the world, but the actual things that are getting made are kind of messed up or different or not working right, doesn't matter how good or accurate your blueprints are. If the proteins aren't really doing the job they're doing, then you can have a whole host of problems. And of course, everything is all tied together, and there are like a million different things that go wrong in aging, and no scientist can focus on anything, on everything. And you have to pick one thing. So I've picked to study, I guess the general way that I would describe all the work that I do is studying proteoform diversity and how protein behavior changes with age. Mm-hmm.
Oh, so fascinating. Let me see if I can repeat it back to you just to know that I got it right.
Yeah.
So when DNA has to go from the blueprint to the actual machine,
yeah,
I can't help myself but do the computer science analog of you go from source code to byte code, or like object code if you want, which is RNA. And sort of like it's the runtime interpretation of the source code, let's call it. And then, but what you're saying is that you'd think that even if you have like sort of something that is maybe not even an order of magnitude, but like say like a multiple of 2 or 4 from DNA to RNA, it really blows up once you go from the runtime, sort of like the object code, to the actual machine because of, and this is where I'm sort of interpreting it, like adding a bit of creative freedom to it, but like environmental factors that lead to the protein behaving in different ways.
Environmental factors certainly play a role, yeah.
But maybe that's my question. You use the phrase post-translational modifications. And so what falls into that bucket? I would just imagine that like it's the conditions that the cell is operating under that makes sort of like these different processes change the function and sort of disposition of these proteins, right? Would you say that's a fair assessment? What else falls into that?
Let me give you an analogy for this, which is, let's say that you take a pottery class and there's a bunch of different, and the teacher hands all 20 students the same bowl. So fundamentally it's a bowl, but you still need to paint it. You need to glaze it. And all 20 of you are going to make a different design on that bowl. And so on some level, they're all bowls. They're still all bowls. They generally have the same shape, but the design on them is dramatically different. And so that's sort of one way that you can think of post-translational modifications, which is protein gets made. Ribosome translates RNA and makes a protein, and then a bunch of other proteins in the cell paint on it. And they can add, they can append sugars to it. They can phosphorylate it. They can stick other stuff onto it. And the stuff that you stick onto it can dramatically change how that protein behaves. So, I mean, so we can return to COVID. We can return to the spike protein, which, is sort of this. Before COVID, nobody would, the general audience wouldn't know what a spike protein was. If you said something about spike protein, nobody would know about it. But I think most of us now can conceptualize what the spike protein is and what it looks like. But what's interesting is that a lot of people haven't necessarily thought about the fact that the spike protein that we may visualize in our head actually is very heavily glycosylated. So it has dozens of sugars that are appended to it. And these sugars can dramatically change the shape and shielding of that spike protein, which is how you get to a state where there are only certain parts of the spike protein that are actually exposed and can be bound by antibodies. And so these post-translational modifications, glycosylation in particular, are tremendously important to the function of proteins in our body and proteins of invading organisms and viruses.
It's so interesting. I mean, we could chat about the spike protein all the time, and just sort of for the whole rest of the interview, just understanding how sophisticated the system of biological penetration is, and how smart, quote unquote, they are at disabling the defense mechanisms of the cell, right? There's another. I mean, I think you bring up glycosylation a lot, and I wanted to know why you picked EIF2 and and glycosylation as the things that you study? What of all the menu, why'd you pick those?
Well, to a degree, it's also because of the labs you join and the expertise of the people around you. And so in grad school, I joined Peter Walter's lab, and he is a biochemist and cell biologist who is principally concerned with biology in the endoplasmic reticulum and protein translation. And they had discovered a small molecule that bound to this protein EIF2B and had really incredible effects on a number of neurological disorders. So traumatic brain injury, for example, where dosage of this molecule really aided in recovery in mouse models. And as of now, there are 4 or 5 different companies that have variants of this small molecule ISRIB in the clinic in various phase 1 trials. Maybe some of them are coming to phase 2 right now in order to treat a whole host of neurological disorders. And so that existed before I joined the lab. A whole wonderful group of scientists did this work. And I thought, okay, well, this is quite interesting. I'm going to further work out the mechanism of action for this drug and for this stress response pathway. And so it was really driven by what's the question that's being asked in that lab, and what do they have the expertise to tackle, particularly as a grad student. You're largely thinking about what is the framework that exists and how do I build on this. It's a challenging task to try to come in and completely make your own thing. Mm-hmm. And in postdoc, in my postdoc, I wanted to be a little bit more on my own, shall we say? Mm-hmm. Or because as a postdoc, you're trying to build independence and develop work that you can then use as a foundation for your own research group. Mm-hmm. And so I interviewed in a few different labs, and I was really excited about the possibility of doing joint work between 2 labs at Stanford. So Tony Wyss-Coray, who I mentioned previously, mm-hmm. who was actually the professor of Solviata, who I did my one of my rotations with, and Carolyn Bertozzi, who is a world-renowned chemist and glycobiologist. And I was really excited about the possibility of working between these 2 groups because aging glycobiology is really understudied because it's really, really hard. So a protein can be glycosylated in many different ways. And my work is on a very narrow type of glycosylation, which is called N-glycosylation, where you're looking at glycans, which are sugars mm-hmm. that are attached to asparagine residues, one of the amino acids. Mm-hmm. And what I was really excited about and why I ended up picking to do this work is because it's really an open area. We've done a lot of work in the aging field of trying to characterize, how RNA levels and to a degree protein levels change with age. And we've gotten more and more and more granular of saying, okay, well, we're going to focus on this organ and then we're going to focus on this region of this organ. And now we're doing it at single cell and even subcellular resolution of saying like these RNAs exist in this cell type at this level, but when it gets old, they're much less abundant or much more abundant. Mm. And I didn't really want to do that. I wanted to take a slightly different approach. And after talking with Tony and Carolyn, one of the approaches that I wanted to take was looking at how protein glycosylation changes with age. And so I'll return to the diversity of proteoforms, which is that if you have one protein and it can have one glycosylation site, sometimes it can have 10 different glycans that are there. And so that can, in reality, be 10 different versions of that protein.
Oof.
And if you have, it gets even more complicated, because if a protein has 2, 3, 4 glycosylation sites, and each of those glycosylation sites can have
10,
5 to 10 different glycans on it, between the combinatorics, you might get into a situation where in a cell, no two proteins are alike, that every single proteoform is actually a unique proteoform because of the diversity of glycosylation. And so that was really appealing to me. And one of the reasons why I wanted to study glycosylation during my postdoc. And the appealing thing is also the really hard thing, because as you can expect, this diversity means that it's very challenging to study glycosylation. But that's what I'm working on.
I mean, it's so interesting. So basically, you study the interaction between sugar and protein, and to very coarse sort of like TLDR version of it. But what you're saying is that proteins have different sites that where sugar can bind to, and not only you could have different kinds of sugar binding to the same site, which may lead to complexity effects to be measured. But if you have more than one site, there may be weird interaction between the sites too, and sort of like non-linearity. Some of them, augment some things, some of them diminish some things. And so it's the composition of the protein ends up becoming extremely different depending on the sugar that binds to it. Is that roughly fair to say?
I think I will roughly, yes. I will throw out a couple caveats. I should clarify. It's not just being bound by the protein, it's being covalently attached to it.
Got it.
So protein and sugar are becoming one and the same.
Right.
So it's
part of the atomic, chemically bound.
And you need to enzymatically cleave it off if you want to remove it. And I should also clarify that it's not immediately clear what different versions of a glycan are relevant and are not relevant, because sometimes you can get into a situation where there's a bunch of different glycans that can exist. Glycans and sugars are sugars, where a bunch of different glycans are attached to a protein. And it really doesn't matter what kind of a glycan is there, so long as a glycan is there. And then you have other instances where the specific glycan that is attached really does dramatically matter. And it's really hard to sift out. This is why it's so challenging. This is one of the reasons why it's so challenging. It's really hard to sift out what differences are relevant and what differences aren't relevant. Because just seeing a difference doesn't in and of itself mean that it's functionally relevant. It would be great if every time you saw something that was different between condition A and condition B, you could be like, oh, it's different, so it must matter. But it doesn't always.
Okay, and so my follow-up question, which is maybe a dummy question, is how prevalent is the phenomenon of glycosylation in protein? So what percentage of cells get exposed to this?
Every cell. So every cell has proteins in it that are glycosylated. And some estimates are that, over 50% of all proteins are glycosylated.
See that? I think
so a lot of proteins are glycosylated
in
some form or another.
And the implication of what you just said before means that essentially more than 50% of the proteins in your body could be essentially unique because of their, like glycosylation profile. Is that fair?
I would say that, in an individual cell, it also depends on the abundance of how abundant a protein is. Because but yes, that what you've said cuts at the heart of what I'm interested in. I don't think I will argue on whether it's everyone is unique or whether sometimes there are two of a variant. But at the end of the day, what we're talking about is in a cell, there is the potential for a tremendous amount of variability of things that we have traditionally thought are the same. That it's, protein A is not always protein A. It's not. There's a whole host of reasons why this thing that we call one name can be profoundly variable. I mean, you can have proteins that exist in the cytosol, and sometimes it can exist in the nucleus. Sometimes it can exist as a dimer with itself, or maybe it exists as a dimer with a different protein. Sometimes it can be tethered to an organelle or free. Sometimes, as I've been talking about, it can be modified with one post-translational modification or another. And so all of these different appendages and behavioral differences can have huge functional consequences. And I think we're really just scratching the surface on this diversity of proteoforms. And I think that's really what I want to dedicate my career to is understanding the diversity of proteoforms in an organism, particularly in the context of how that changes with age, and trying to tease out what of those differences are functionally relevant and how we can actually modulate those differences to restore some degree of the youthful state to an old organism. I think that's the dream. Studying differences between old and young just for the sake of studying differences is exciting, but it's, I would say that I won't speak for everyone, but for me, and I think a lot of scientists, you want your research to have an impact. You want your research to not just make people, have more knowledge. You want to help people's lives get better. You want people to live longer, healthier lives. You want to contribute to the generation of medicines that improve things.
No, 100%. There's an underlying thread there that I think is beautiful and empowering about how, if you think about, you as an entity, or like the proverbial you, the thing that you think you are as an entity, and the fact that every subdivision of you, like the cells and the proteins within the cells, even though they're made from the same instruction set, there's still like room for the concept of uniqueness, and that like every cell, every little part of you is doing its own little thing toward the project of being you. I think there's something really cool there that like maps to I think something that I feel in society, where people feel disconnected from their role in society. And I think that there's like a cool thing there where it's like a room for uniqueness and the fact that the set of environmental factors that made you who you are is because you're part of a larger social organism that is uniquely glycolated, or like, incentivized by sweetness. How about that? To behave in certain ways, right? And I think that there's something really cool there. There's another layer too, which I want to unpack a little. That is, you mentioned that you really did not enjoy the mouse work. Now, I'm 100% with you on the fact that I get weird about doing stuff to living creatures. But I'd love to hear your perspective on it, knowing that you wanted to be a scientist as a kid.
Yeah.
And that's sort of like, in my mind, this very archetypical thing is like scientists are in labs with mice.
Yeah. Well, yeah. It wasn't, I should say that as a kid, it wasn't necessarily always biology. Thankfully, physicists don't need mice to do their work, nor do chemists. And in high school, I every opportunity that I had to do work with organisms or with fetal pigs that have, for dissection, all this stuff, I turned away from it as much as I could, because on a personal level, I found it unpleasant and upsetting. And it is, because it is. But when I was deciding on what I wanted to do in grad school, I kicked that decision down the road. And I didn't do mouse work in undergrad or grad school. But during my postdoc, I had a long think about this, and I've done aging research in C. elegans, and I enjoyed it quite immensely. But I thought, basically, do I want to do this career? Do I think that this is a worthwhile endeavor? And do I think that this is important to do? And the answer to that was yes. And then the follow-up question was, do I think it's possible to do this without doing animal research? And I think the answer to that is no. So I think if you want to do medical research, there needs to be, given our current levels of technology and what we can do, there needs to be animal work, as painful as it is. It's very painful for me to do the work, but you have to do it with the utmost respect for the sacrifices that the animals are making. And you have to do your damnedest to do the work with as few animals as possible, and to make the most of the animals that you use. It's like when people talk about using every part of the buffalo or these sorts of things. I try to bank as many tissues as I can, and to use as few mice as possible in an experiment that I think will prove, if I think this will be sufficient, I try not to go overboard. It's painful. It's tough. I don't like it. I've been doing it for 2 years, and I still don't like it on a emotional level. But I do understand that it is necessary. It's hard.
No, but it's refreshing to hear your perspective, Michael, because it's easy to get detached from this stuff too. As a consumer of something, you consume medicine, and you never even take into account the process of the discovery and distribution, the whole chain of events that led to you enjoying the benefit of something, of all the life that it took and all the suffering that it took. And so I think it's really important to highlight a perspective like yours, somebody who has dedicated their life to the pursuit of something as gigantic as, aging itself and, warding off death, that even somebody who dedicates their life to this, this is an impactful thing for them, or this is something that impacts them terribly. And I think that's easy to overlook. So I greatly appreciate that perspective. And, I chickened out from the whole endeavor very early on. I mean, I couldn't do medicine for the life of me because the whole thing is just too much for me. The front line in the battle between life and death is, I'm not cut out for that. I mean, and I have all of the respect in the world for people who go into that and sort of face it head on. But I think it's important to realize that you need sort of the nuance and the ethical decision of even though it may be painful, it still needs to be done. And sort of that wrestling of the soul figuring out this stuff, I think is just important to note. Let's get a little bit weirder with it, which is the ethical considerations of the success of your project, right? So what does a world where aging means something different because of your contributions look like for you?
Yeah. Well, I think there's two ways of thinking about that. There's what is what is realistically going to happen and what is the, sci-fi version, right? So like the realistic version is through lifestyle changes and therapeutics, people's lives are extended by, some 10 to 15% on average, and they generally live healthier. They're just living longer. And so in that world, it's probably fairly similar to what we've, been seeing right now in the developing world, or where people just generally live longer. In both the developed and the developing world, particularly when you look at the developing world and shifting into the developed world, lifespans increasing, it's generally a good thing. And healthcare costs, if you can increase lifespan without increasing the period of frailty, shouldn't really go up. Indeed, the ideal therapeutic is making the period of frailty shorter while still extending lifespan. And in those cases, you would hope that healthcare costs would go down. Of course, the sci-fi version is everybody lives forever. And there's a bunch and the even more sci-fi version is everybody lives forever and gets turned into computers. So I don't necessarily think that the sci-fi versions are going to happen in our lifetime. I certainly would like to be wrong, but I think the realistic scenario of the impacts on aging work are largely positive. And that I think a lot of the concerns that people might have of access only for the rich, or, people living to 200 and being infirmed for the last 100 years. I don't think those are going to happen. I don't think the system in the next 50 years is going to be that greatly different as a result of aging work, frankly, because I think therapeutics are so far away. I think the best thing that people can do is just actions that are generally smart, of eating well and sleeping well and exercising, which is just, you'll be healthier in general and you'll also live longer. So that's the best thing you can do.
I hear you on a couple of notes, because this is the thing that I've been thinking about with the advent of AI and sort of like my thinking around aging is that the sci-fi scenario is just taking the limit to infinity of the project that we're on now, right? That is, let's say it's 15% longer, like more longevity. But you could also think of it like as an engineering challenge where it's like, all right, 3% improvement per year, or it gets longer by 3% every X amount of time. And that compounds over time. And so, it changes the dynamics of society, and it changes like the age at which you change. It changes our notion of social institutions, right? Like if you're going to be around till 150, what age do you get married at? And so all these different types of implications come from longer lifespans that I think are super interesting. And I agree with you that I think they're, by and large, very positive. But I think that it's really interesting that I've had a harder time dividing the realistic case from the sci-fi case because of things like AI, where that was sci-fi 5 years ago. When we were undergrads, the level of AI that we have today, at least in the conversational sense, felt like, this is 20 years out. And not to say that progress in AI will map onto this thing at all, but AI will most likely serve as an accelerant to this type of research, this type of, it'll help you crawl the search space of possible things in a way that makes it faster. And I think that as humans, we navigate a world where we sort of have a baseline of how society functions, how biology functions, how institutions function. And with changes like AI, but not just AI. I think just the times in general. I mean, like biological weapons and nuclear weapons and new sources of energy and like all these insane things that are going on now. I think that like when I get very interested in like the possibility of or something like aging research, having license to actually, okay, this is a project that we're embarking on, that we're going somewhere, and what does that somewhere mean for society? So I think it's fun to hear your thoughts on that, because, it's very different to see it from your perspective, where you're like, okay, this is what a molecule does to the function of a protein. But that maps to, the concept of, how long is it the educational period? How long are you in school for? If, you're going to be around for 200 years. So do you, how much runway do you give yourself when thinking about where this goes?
Okay, let's do it. Let's do the runway. I like sci-fi, so let's do it. I'm down.
Hell yeah.
I'll preface this by saying I don't necessarily think that the sci-fi, as I've said, I don't think the sci-fi version of aging is going to happen, but let's go. I think it really depends on the specifics, because you can get real weird, right? Mm-hmm. You could imagine a scenario where, again, imagining, so we can imagine whatever we want. If your lifespan extension is also expanding your period of fertility, then people are probably going to get married later and have kids later. I mean, we've already seen that marriage and kids is happening later and later. I'm 30, and the only kids I have are my cats, Ada and Dalton. I needed to put a plug in for them somewhere. And my grandma had 9 kids starting in her early 20s. Times have changed. Of course, there's a limit at a certain point of how old you can be if you're a woman, how old you can generally be when you have kids. But you can certainly imagine that if lifespan extending therapies or fertility extending therapies exist, that, as we've already seen based on other trends, that this expands. You'd mentioned education. That the time of training has expanded as well. A lot of people weren't going to college. Now a lot of people do go to college. It was less common for people to get graduate degrees. More and more people are getting graduate degrees. So I think as the knowledge base increases and as the skills required to be really good at something keeps increasing, there's an arms race of how long you need to train for. I mean, I have friends who are going to be, various specialty surgeons and doctors who they have to go to med school, then they have to go to residency, then they have to do multiple types of residency, they have to do internships, and really they're only becoming a neurosurgeon when they're in their, I don't know, maybe late 30s or 40s. That's not my field, but it takes a while. In the biological sciences, it's expanded dramatically. Like PhDs used to be 4-ish years. Now they're stretching into 6 years in the US. Postdocs used to be a couple of years. Now they're stretching into, 3 to 6 years. And so in the biological sciences, when there are grants for young investigators, they're in their 40s. Like it's young investigators are 40-year-olds. I mean, I think the general trend with everything in society is things are getting stretched out and expanded. And you can imagine that this would really kick into overdrive to a degree with some therapeutics. But at the same time, if you're not slowing brain development with any of this stuff, then somebody is cognitively going to become an adult more or less at the same time in that made-up scenario. Of course, you can get into weird sci-fi things of if we all become robots, then, we're not even having human kids anymore. So
Yeah. I mean, like, yeah, that's the next level of
it can get real weird real quick.
But things like I get really interested with like how it changes our relationship with the inevitable things in life, like, and like maybe that's like it's just me chickening out from saying death. So if life is more abundant and longer and more fulfilling and richer and healthier, then the tragedy of death is magnified, right? Like it just, if somebody dies at, I don't know, if lifespans are 150 and somebody dies at 70, that's a tragedy. It's like, they died young. And so but I think that's a difficult question to wrestle with. I think like it took, an immense amount of effort to me to figure out how should I feel about this stuff. And that this is, in all honesty, this is the project that we're embarked on as a species. Like we're just trying to defeat the thing that makes it all stop. But what does it mean when we do? And what does it mean when we get a significant victory? And I think, this is stuff that I think is important to think about every now and then. Just as a reminder of like, what you get when you go there. There's another thing that you said that I thought was really interesting in the world of grad school and PhDs and academia in general. I think in a world where more advanced study becomes more commonplace, it will add to, I think, already a difficulty in academia today. That is the explosion of literature that PhDs have to sift through and comb and organize and rank and cite and find their lane in, which is like, it's a very daunting, difficult task. This is very difficult material to read, and it becomes incredibly jargony and lingo-y and insidery at the higher up the tower you climb. Can you imagine how weird that gets when like 60% of the population is doing that? First of all, let's unpack the first problem as it is today, and then how do you feel we should or would navigate it later on?
Oh yeah, I don't know how in that scenario, I don't even know how you navigate it. You just get more and more siloed, which can be problematic because it becomes only possible really to read so much a day and to be knowledgeable about so much. And so when the scope of knowledge keeps increasing, you have to pick a narrower and narrower and narrower thing to be knowledgeable on. But I think another scenario is potentially, how physics sort of has gone, where it gets more and more and more collaborative in many ways because the questions are so big that you need more and more people. And so it actually isn't the end of the world if people are super specialized in one thing, when you have a really big team and Jim over here is the expert on this and Jenny over here is the expert on this. And so I think probably the way forward is just better communication and teamwork when you get into a scenario where it takes so long to be an expert on such a niche topic, and probably augmented by technology as well. Which is already what we have. It used to be that you had to go to the library and physically flip through the journals in order to find the things that were interesting to you. Now I have PubMed alerts for keywords. And so I get articles sent to me that are any article that's sent to me that has, aging and glycosylation in the abstract. So you can, of course, augment with technology. And I imagine just like the arms race between cells and viruses, there's, an ever-present arms race between our lack of knowledge and the scope of the field, and it'll keep going. And we'll keep figuring out ways to eke out more and more ability to do things. And I think it'll probably end up with more specialization in bigger teams. for biology.
Yeah, it's exciting. I mean, I think that the project that you're embarked in, that we as a society are embarked in, is so marvelous. I think it's just like it's a thing that, like, if I think about it too much, it just really contextualizes how tiny I feel and how special I feel, and I think it's awesome that, personally, to be able to talk with you about, like, you're in the weeds doing the actual work and, like, actually measuring the stuff that, you abstract time, you map time out. And, like, for the case of the nanobody paper, it actually has therapeutic effects already. So, like, it's proven that, like, that this is like, well, it's directed toward therapy, and it's directed toward interventions with humans and actually sort of extending their lifespan. What are some of the things that get you the most excited about your work when thinking about how it will apply to humans over time?
Honestly, one of the things that gets me most excited is the day-to-day interactions with people. I really love working with. I have a collaborator that I work with quite extensively on all my projects. This guy Johannes, who's wonderful. I get really excited to talk with him about science. I get excited to talk with the undergrads and master's students and rotation students that I work with. I really get a kick out of doing the science with people, which is why I think when we move towards bigger teams, it'll probably be for the better. And I also really am excited by, and I think anybody who does research has to be excited about this, the thought that when you discover something truly new, you're the only person in the world who knows this. That, like, when you reach a certain level of granularity and newness, nobody else knows this for sure. Well, you don't really know it for sure, but no, you can piece enough things together where you're like, okay, I'm the only person in the world who knows XYZ. That's pretty damn cool. Let me tell people about it. And then they can become the only person to know YZX because my discovery led them to have some insight in their own thing. And now you're just building off of each other, and everybody's thinking, oh, wow, there's this new thing that I didn't know because this other person then created this new thing that I didn't know. So I think unlocking puzzles to the universe is the big picture science thing that has always excited me and why I wanted to do science, which is, doing new things and learning new things. It's really cool. It's really cool.
Amazing. What a way to wrap it up. Thank you so much, Michael. I really appreciate the time. This was great. I hope we get to do it again. Yeah, you got it.